Hi everyone, welcome back to my channel. Today's topic is stability testing in pharma, zones, conditions guidelines. What is stability testing? Stability testing is a critical aspect of pharmaceutical product development. It provides evidence on how the quality of a drug substance or drug product varies with time under the influence of environmental factors such as temperature, humidity, and light. This helps in determining the shelf life, storage conditions, and packaging of pharmaceutical products. Objectives of stability testing: to ensure the drug maintains its identity, strength, quality, and purity throughout its shelf life, to establish recommended storage conditions, to determine expiration dates and retest periods, to understand degradation pathways and stability-related risks. Types of stability studies. 1. Long-term stability testing, conducted over the proposed shelf life of the product under recommended storage conditions. 2. Accelerated stability testing, done under stressed conditions, higher temperature and humidity, to predict long-term stability. 3. Intermediate stability testing, required if accelerated testing results show significant changes. 4. Stress testing, helps identify likely degradation products and mechanisms. Stability zones, the world is divided into climatic zones based on temperature and humidity for the purpose of stability testing. Zone I, temperate climate, Northern Europe, Canada. Zone II, subtropical and Mediterranean climate, Southern Europe, Japan. Zone III, hot and dry climate, Egypt, Saudi Arabia. Zone EVA, hot and humid climate, ASEAN countries, India. Some areas. Zone IVB, hot and very humid climate, Indonesia, Nigeria, coastal India. Standard storage conditions, ICH guidelines. The International Council for Harmonization, ICH, provides detailed guidance, ICH Q1A, Q1F, on conditions for stability testing. Below are the standard storage conditions. Long-term stability testing, Zone I2, 25 degrees Celsius plus or minus 2 degrees Celsius / 60% RH plus or minus 5%, Zone 3, 30 degrees Celsius plus or minus 2 degrees Celsius / 35% RH plus or minus 5%, Zone EVA, 30 degrees Celsius plus or minus 2 degrees Celsius / 65% RH plus or minus 5%, Zone IVB, 30 degrees Celsius plus or minus 2 degrees Celsius / 75% RH plus or minus 5%, accelerated stability testing, all zones, 40 degrees Celsius plus or minus 2 degrees Celsius / 75% RH plus or minus 5%, usually for 6 months, intermediate conditions, if needed. 30 degrees Celsius plus or minus 2 degrees Celsius / 65% RH plus or minus 5%, duration of stability studies, long-term testing, typically 12 to 60 months, accelerated testing, 6 months, intermediate testing, 6 to 12 months, if applicable. Key regulatory guidelines, ICH Q1A, R2 stability testing of new drug substances and products, ICH Q1B, photostability testing, ICH Q1C, stability testing for new dosage forms. ICH Q1D, bracketing and matrixing designs, ICH Q1E, evaluation for stability data, ICH Q1F, stability data package for registration in climatic zones 3-4, replaced by WHO guidance in some cases. Important parameters tested, assay, potency, degradation products, physical characteristics, appearance, color, odor, dissolution, for solid dosage forms, pH, for liquids, microbial limits, moisture content, container closure integrity. Conclusion, stability testing is vital for ensuring the quality, safety, and efficacy of pharmaceutical products. By complying with global regulatory guidelines and selecting appropriate testing zones and conditions, pharmaceutical companies can confidently establish shelf life and storage requirements that ensure patient safety and product reliability. If you like the information, then like, share and subscribe. Thank you for watching.